Hi, welcome back to chapter 5. So in part 1 lecture video, we talked about monomers and polymers. We talked about how we form them, that we have dehydration or hydrolysis reactions. And we talked about carbohydrates, what the formula for carbohydrates are, what carbohydrates are used for. We talked about their monomers. We talked about storage or structural carbohydrates. We're going to pick up our discussion of biomolecules here by talking about lipids. Lipids typically, well, according to your textbook, they don't have polymers. Some will make a case that fatty acids and glycerol are monomers, but according to your textbook, they are not monomers, so we'll stick with that. So they don't really have this particular monomer. They don't have a particular chemical feature that defines them. Really, what the unifying feature of lipids is they do not mix with water. Do you remember what that word is? Lipids are hydrophobic. And the reason lipids are hydrophobic is because hydrocarbons are nonpolar. So all these carbons and hydrogens, the electrons are floating equally through this whole molecule. You don't have any delta positives or delta negatives that would really want to interact with water. So fatty acids, all lipids, this would be fats, oils, waxes, they don't mix with water. The major biological groups of lipids are going to be fats, phospholipids, and steroids. And these three biological groups are going to have related structures, but pretty diverse structures, honestly, and pretty diverse jobs they're going to do. Here you can see that when you are creating a fatty acid, or when you're creating a triacylglycerol, a fat molecule, you start with glycerol, and it has these 3 hydroxides on it. Each of these hydroxides can undergo this dehydration to synthesize a bond between glycerol and the fatty acid. And this is called an ester linkage. This hydroxide here is going to go through a similar reaction and end up with another fatty acid chain. And this hydroxide can go through another dehydration to come up with another fatty acid chain, giving you these 3 ester linkages. And all 3 of these chains are nonpolar, and so this fat molecule is not going to mix with water. Fats, when we're talking about just a regular fat molecule, they have 3 main functions. One is energy storage. So if you are eating a food that has a lot of fat in it, it is going to have a lot of calories in it, because fat is one of the most space efficient ways to store energy. And then after you've eaten that, if you haven't burned that energy, if you don't use up those calories, your body is going to store a lot of that energy in your body. So you store a little bit of energy as sugar. We already talked about that. But sugar, it would take a whole stinking lot of space to store all your extra energy as sugar. So you store most of your energy as fat. By doing that, it condenses the amount of space it takes, so you are able to move around. Things like plants that don't care if they're able to move around don't really worry about storing things as fats as much. Fats also create a lot of insulation. So, pretty soon it'll be winter and you want to stay warm. Your fatty covering around you helps you stay warm. If you think of animals like seals or a walrus that lives, in more polar or water areas, they need a lot of insulation to stay warm. And they get that insulation through blubber, through a type of fat. Your fats also pad and cushion the body. So every time something pokes you, it's not going to go right straight through one of your body organs. It's not going to cause a lot of damage to you because of that layer of fat around your body that pads you and cushions you and protects your vital organs from being damaged. Fats are constructed from glycerol and fatty acids. So glycerol is this 3-carbon alcohol. It's got these 3 hydroxyl groups. And a fatty acid is a long carbon chain. Fatty acids can vary in how many carbons are on them. And they'll have different names. So this This fatty acid is called palmitic acid, but your fatty acid will combine with the glycerol in a dehydration reaction. Let's watch this video about it.
Butter, lard, margarine, and salad oil are composed of lipids called fats. To make a fat molecule, 3 fatty acids bind to a molecule of glycerol. For this reason, fat molecules are technically called triglycerides. Fat molecules do not mix with water because they have 3 long nonpolar hydrocarbon tails. Cells use fats for energy storage because the tails hold more potential energy than other biological molecules. The fatty acid tails in a fat can vary in length. A very important characteristic of fats is the number of double bonds in the hydrocarbon tails. Fats that contain only single bonds are called saturated fats. Fats containing double bonds in 1 or more tails are called unsaturated fats. Unsaturated fats tend to be liquid at room temperature. Vegetable oils are examples. Saturated fats like butter and lard are solid at room temperature. Saturated fats in the diet can lead to heart disease, while unsaturated fats are safer.
Okay, so this described saturated and unsaturated fats. We haven't talked about that yet. We'll get back to that in just a second. So fats will separate from water because water molecules will hydrogen bond with each other, and they're not going to hydrogen bond with fats. So the water and the fat kind of separate from each other. In fat, you have 3 fatty acids. So 1, 2, 3 joined to this glycerol backbone. And so this makes it called a triacyl. So these triacyl glycerol, or sometimes it's called a triglyceride. And when you're making your fatty acids, they can be different lengths, they can be different kinds. You can have 1, 2, or 3 different kinds of fatty acids in your fat molecule. So fatty acids vary in length, and also they can vary in the location and number of double bonds. So when you have a saturated fatty acid, you have as much hydrogen can possibly be in there. It is saturated full with hydrogen. In an unsaturated fatty acid, you've taken out some of that hydrogen and replaced it with a double bond. So these carbons, instead of having hydrogens coming off of it, they have this double bond here. You can notice that a saturated fatty acid is a nice straight line. So these fat molecules are going to pack very tightly together. They can be nice and efficiently packed together, and they will form typically solids at room temperature. When you throw in a double bond, it kind of forms this little kink. So this starts going off to the side here. So you can see this bending in this unsaturated fatty acid. Because of those, these fatty acids are not going to bond together quite as closely to each other. And so because they're not quite as close, they're a little bit more able to slip past each other. These are going to typically be liquids at room temperatures. So saturated fats are typically animal sourced fats, and unsaturated fats are typically plant sourced fats like vegetable oil or olive oil. Okay, let's see, we just said this. Fats made from saturated fatty acids are saturated fats and solid at room temperature. They're mostly animals. And fish fats also are unsaturated, and they're liquids because of that kink that doesn't allow them to get quite so solid next to each other. The type of fat you're eating can make a big difference in your health. So if you're eating a lot of fatty acids, if you're eating a lot of saturated fat, it can contribute to plaque deposits. So that means you're going to get stuff lining the inside of your blood vessels, and that then can lead to cardiovascular disease. So back, 30 or so years ago, people thought, well, why don't we take unsaturated fats that don't cause heart disease and use unsaturated fats? We'll just make them solid at room temperature by hydrogenating them and come up with hydrogenated fatty acids. And maybe it'll still be a plant oil, so it should be healthier. And so they created things like margarine. Today we call them trans double bonds. And pretty quickly we learned trans fats are even worse than saturated fats when it comes to heart disease. So trans fats contribute to cardiovascular disease even more. So things like Crisco and margarine that were very popular throughout the 80s. Now we know that these fats shouldn't be used. And if you buy baked goods or processed foods, it shouldn't have any trans fats in it anymore. Okay, the next type of fatty acid, or the next type of lipid, we're going to talk about are phospholipids. So a phospholipid has taken this glycerol head group and it has two fatty acid tails, but this third area, instead of adding that third tail that we just saw, it's going to replace it with a phosphate group. This is going to end up with a molecule that has a hydrophilic area, so that phosphate group has a charge on it. That is going to interact with water very well. And a hydrophobic area, these two tails that do not react with water. So you have two fatty acid tails that are hydrophobic. Then the head is going to be your phosphate group. And it typically also has choline, and then this glycerol is considered part of the head. You can also see you can still have saturated or unsaturated, so straight or kinked tails when we're talking about these fatty acids. So this is going to react well with water, and these are not going to react water. That means that you can put together a bunch of these phospholipids together, and they automatically, when you throw them into water, will form these bilayer. The bilayer is going to have all the hydrophilic heads happily pointing up towards the water on both sides, but these fatty acid tails are hydrophobic. They're going to hide away from water. So the inside surface of a cell membrane and the outside surface of a cell membrane are hydrophilic with those phospholipid heads, but the inner part of a cell membrane is hydrophobic with those tails. And cells are able to exist because of this structure of phospholipids. So your cell can have a watery environment inside and a watery environment outside, but water isn't able to really pass through here unless you put in a channel or something. So this is really good at giving integrity to what is able to go in and out of the cell. And it also means that this cell is not going to be dissolved because the phospholipids, or the fatty tails, are not going to be interacting with water to allow those phospholipids to be dissolved. Okay, our last category of fats are steroids. Steroids are lipids that have this 4 rings in them. We have 3 6-hydrocarbon rings and 1 5-hydrocarbon ring. Sometimes you'll hear them called, 3 beehives in a doghouse or something like that. Different steroids will have different functional groups on it. So here you can see a hydroxide functional group. You can see a couple of methyl functional groups. One type of steroid is cholesterol. Cholesterol is a part of animal cell membranes. Cholesterol is important in cell membranes because it will get into the phospholipids. Let's go back. So it'll just slip itself into the middle of phospholipids and prevent those phospholipids from getting too close together. So it keeps your cell membranes liquid instead of freezing solid. Cholesterol is also used to make other types of steroids. If your body naturally makes cholesterol, you can also eat cholesterol, but if you have a diet too high in cholesterol and get too much cholesterol in your blood, it could cause plaques and also contribute to cardiovascular disease. Let's pick up proteins and DNA in our last part of this video. I'll see you back.